Hi everyone. Today we are going to continue with the cell cycle and some of the parts of the DNA synthesis, protein synthesis, which comes under chapter 3. So cell cycle is can defined as series of changes a cell undergoes from the time that it's formed until it reproduces. And lots of cells goes under cell cycle. Only some cells, once they differentiated, they don't no longer going through the cell cycle process. With related to the process, there are two major periods of cell cycle. That is the interphase, where the cell grows and carries on its usual activities, and the cell division process, where the mitotic phase, where the cells divides into two. So if you look at this figure, you would see that the green portion is all comes as the interphase, and it's going to further divide it into three parts. We are going to see it in later. And look at the direction. It's going in one direction all the way up to here. And then you would find the mitotic phase, which also included in two parts, the mitosis part and the cytokinesis. And it will circle, so that's why it's a cycle. It will keep on happening in the dividing cells. The subphases of the interphase would be the gap1 phase, the synthetic phase, and the gap2 phase. The gap1 phase you might encounter in some books as growth 1 phase, or the gap2 as the growth 2 phase. Doesn't matter, it's the same meaning. In the gap1 phase, there's vigorous growth and metabolism happening. So lots of organelles are doubled, lots of proteins are synthesized for the new cell. So lots of things happening. But cells which are permanently ceased dividing, has stopped dividing, are said to be in the G0 phase rather than going to the gap1 phase. For instance, like some nerve cells, not all nerve cells, certain nerve cells, they do in G0 phase. The S phase, S stands for synthesis. What does it synthesize is the DNA. So the DNA replication is happening. So we have to see some of the DNA replication process in this course. The gap2 is the preparation of the cell division. With related to the DNA replication, the cell, to give its copy, it has to make another copy of its blueprint. So that is where the DNA replication happens. And the double-stranded DNA is in twisted form. So to make another copy, you have to make it unwind or unzip it. So there are two things happening where at the replication fork, the point where the DNA stands starts to separate, and you get a replication bubble, where the active area of replication happening at that time, because not suddenly the DNA strand open it up, it's gradually open it up, unwinding, so the bubble moves on, and the fork is where the starting point. Each DNA strand, of course, now you know it's a double-stranded. Each strand works as a template for the new complementary strand. So at the end, you get, in the new strand, you get one old strand and one new strand. So it's not fully new. Therefore, we called it as the semi-conservative replication. The RNA plays a role in the DNA replication process because the RNA laid down its small subunits, we call them as primers, to start the DNA replication process. And there are some enzymes involved. These are very good MCQ points. DNA polymerase is the one which attaches the primer, the RNA segments, to the DNA template, and it begins adding nucleotides to form the new DNA strand. And DNA polymerase, it synthesizes both new strands at the same time. So you would get one strand as a leading strand, which is the one which is formed continuously, and the other strand is formed as segments, and then later they're going to combine together. We call it as the lagging strand. So DNA polymerase, it works in one direction. The leading strand formed continuously, the lagging strand forms backwards, and then it formed discontinuously, and at the end the DNA ligase involved with combining these lagging strands together. So here, before that, the end result of the interphase would be producing two identical daughter DNA molecules, and during this mitotic cell division, one complete copy will be given to a new cell. So as I mentioned before, each new strand has one old strand. Therefore, that is why we call it as the semi-conservative. Sorry for that. So here it shows the replication, and here the replication fork, remember where it starts, and this area is called the replication bubble, where the active DNA replication is happening, and the DNA polymerase is moving. At the same time, the forward strand is the leading strand. It's formed continuously, and you would see the lagging strand, it forms not continuously. These segments formed here, then another segments would form here, and then later DNA ligase comes and bind into it. With related to the other part of the cell cycle would be the mitotic phase, which includes mitosis and cytokinesis. And in the mitosis or the M phase, it's the nuclear division is happening, and it involves four phases, which is the prophase, metaphase, anaphase, and telophase. And you can remember them as PMAT, taking all the first letters of these four phases. And if you look at the key features of what is happening with each phase, it's very easy to remember. So prophase, we divide them into two major parts, early prophase and late prophase. So what will happen with early prophase is that before the nuclear division starts, the chromatin, remember the three combinations, DNA, nucleosomes, and RNA, they're very loosely arranged. It's like dispersed all around the nucleus. So they start to condense, and then at one point the centromere is holding up these duplicated chromosomes, because by that time one chromosomes, they have doubled up, and once they condensed, you started to see the actual chromosomes, and you do see the chromosomes have been doubled up, and it's held at one point. It's like a little bow, and the center of the bow would be the centromere. And the centrosomes, as it duplicates, they begin synthesizing microtubules, and they push each centrosomes to opposite poles of the cell. So the centromere is the one which holds up the duplicated chromosomes. But remember the centrosome, which is like the right angular structure outside of the nucleus, those starts to move on to either sides of the cell, like the cell poles. Imagine it's like a globe, and the satellite, two satellites at right angle outside the globe, and these two satellites starts to move to both sides of the north, to the both sides, both poles, which is one for the North Pole and one for the South Pole. And as they move, they form some little strands, which is like microtubules. So that's the best example I can give it to remember. So the strands which you use, which they create, is called the mitotic spindle. And So this is what I mentioned. Here, the centrosomes, they also doubled up. So these two structures goes to one side, one pole, and these two structures goes to the other pole. While they're moving, they form the mitotic spindle. And from the centriole itself, there are short fibers. They are called the asters over here. And you would see that the centromere is attached into the middle, and you see duplicated chromosomes. We give them a special name called the sister chromatids. So what will happen at the early prophase is that this is the two centrosomes starts to go outside to the either poles. The central chromosome starts to condense and they started to appear. And mind you, you would say the nuclear membrane is still intact, which means like it has not dissolved yet in the early prophase. The late prophase, you see the starting of dissolving of the nuclear membrane, and you find here there's no nuclear membrane over here. And the special microtubules, they attach to the specific area of the centrosomes. We call them as the kinetochores. See the kinetochores over here, and they started to and they just attachment is there. Nothing has been happened until yet. No pulling has been happened. It's just the attachment. Then at the metaphase, it's very easy. It's just like central chromosomes have been arranged in a metaphase plate. It's like the chromosomes all arranged at the equator line of the world. It's like that. And during the anaphase, the separation of the sister chromatids happens. The mitotic spindle pulls the chromosomes into either side. So therefore, the sister chromatid separates. And because of the pulling force, they take a V shape. These chromosomes goes to this side. These chromosomes goes to this side during the anaphase. And at the telophase, and the cytokinesis happens together at the end of the telophase. If you look at the cell cycle, you would see towards the end of the telophase, the cytokinesis process almost started to happen, where you see a contractile ring or cleavage furrow is formed between these separated chromosomes, and you started to see nuclear envelope is started to form again, and then chromosomes are surrounded by this nuclear membrane once again. So it's like you take a big balloon, try to squeeze from the middle. That's the cleavage furrow which is happening at the cytokinesis process. So like everything is being controlled tightly. So there is a control for the cell division as well. There are signals to say, okay, go ahead and start cell division. And there are signals to say, okay, stop cell division. So we call like go signals, which stimulate the cell division, stop signals, which inhibits the cell division. So the go signals would be the critical surface to volume ratio of the cell. When there is area of membranes becomes inadequate for exchange of chemicals, exchange of certain stuff, then you may require for the cell division process, like for injury, like a tissue damage. So you have less amount of cells available for the surface exchange, so you need to have more surface. So that's why you have to divide the cells and make that area repair and then have the proper function back. The chemicals, certain growth factors, hormones involved with the go signals. Also, stop signals. When the cells start to divide, it can't keep dividing. If there is a gap and if it's been filled, the cell-to-cell contact involved with this process, and they signal it and they tell like, okay, now it's time to stop it. So we call it as the contact inhibition. And also, there are two crucial proteins available to accomplish the synthesis phase during the entire mitosis, which are the cyclins and CDKs. And CDK stands for cyclin-dependent kinases. They depend on the cyclins itself. So cyclins, they are the regulatory proteins and they accumulated during the interphase. And the CDKs, they activate the cyclins when they bind to them. So cyclin CDK complex in turn activate enzymes cascades, and that will prepare for cell division. Enzyme cascade is like you have enzyme A, B, C, D. So enzyme A needs to be activated. Activated enzyme A is required for the proper function of B. Then when the B is activated, then the C is going to activate. So everything is being linked together. So that's called a cascade. And cyclins, when they destroyed after, they do not exist as it is. Once their function is done, when the cell division is done, then they destroyed by several processes itself. So if the processes needed to be happened, then cyclins is being produced again. Also, there are several checkpoints during this cell cycle, which are the key events of the cell cycle division, because if you cannot pass these checkpoints, then cell won't divide. So G1 checkpoint, G2 checkpoint, and the spindle checkpoints are the three checkpoints that you encounter in the cell cycle process, which is the G1 checkpoint is near the end of the G1 phase, G2 checkpoint is near the end of the G2 phase, and the spindle checkpoint is actually at the M phase, and it's especially at the metaphase anaphase transition time. And out of those three, G1 checkpoint, we call it as the restriction point, is the most important of three major checkpoints. And if a cell does not pass the G1 checkpoint, it enters to G0 and it will no longer divide again. And with related to the cell cycle, because we are talking about it, and cell cycle involve lots of protein synthesis, we have to discuss a little bit of protein synthesis in this chapter as well. So we know DNA is the master blueprint for everything, and it holds and code for the protein synthesis as well, because if you think of we made from lots of structural proteins, we have enzymes made from proteins. So lots of things is due to proteins. And then DNA have the message or the code for how to make these proteins. And how it does is first it transcribed and then it's translate. So when we take the whole DNA, it's composed of lots of genes. So a segment of the DNA which codes for one polypeptide. I hope you remember what is a polypeptide. It's a combination of several amino acids. It's a linear structure. A segment which codes for one polypeptide is referred to as a gene. So the code is determined of the DNA is by its nitrogen bases. So what are the nitrogen bases? Adenine, guanine, thymine, and cytosine. And the code actually consists of three sequential bases. So we call them as triplet code. For example, you would see, you can find GGC, guanine, and cytosine, and it can codes for the amino acid proline. And if you have the GCC combination, this triplet code, it could code for the arginine amino acid. So based on which triplet code you code for different types of amino acids. And also, when you take a gene, it's not all the segments of all the DNA, all the nucleotides arrangement would not code for protein synthesis. There are places called exons there are places called introns. So exons are the ones which code for amino acids, and that is what is important. But introns, they do not code for any amino acids, they're just intervening segments. So before we talk about the protein synthesis, because lots of other molecular structures involved, we have to know what are they. So RNA is type of molecule which is involved with the process of the DNA and the protein synthesis. So what does RNA do is, the RNA, they copy the DNA code in the nucleus and it carries into the cytoplasm and to the ribosome. But all RNAs, they formed in the nucleus. And the RNA, it could differ from the DNA based on its nitrogen base, uracil, because in DNA you have thymine, but RNA you have uracil. And also, instead of deoxyribose sugar, RNA does have the ribose sugar. There are three types of RNAs. They are the messenger RNA, the ribosomal RNA, and the transfer RNA. And if you look at the first letters, that is what it's been mentioned with the lowercase letter in those RNAs. So messenger RNA is a single-stranded RNA, and it codes from DNA template strand and is copied with a complementary base pair, resulting in a strand of the mRNA. And this process, we call it as the transcription. You transcribed the DNA into a mRNA molecule. And the mRNA, it does keep the triplet code of the DNA. And what will happen with the ribosomal RNAs is the structural component of the ribosome, and that is where the protein synthesis happens. And it would, along with the tRNA, it helps to translate the message which is carried by the mRNA molecule. The third type is the tRNA molecule, which is a little bit of awkward in shape. Actually, it does have loops. And it, in one point, in the 3' end, it carries the amino acid component, and it does have an anticodon for the codon which you find for the mRNA. And it has special areas which contain the specific triplet code, that is the anticodon, which allows each tRNA to carry only a specific amino acid. So based on this one, you would carry a specific amino acid tRNA. So anticodon of the tRNA, it's going to complement with the base pair with the codon of the mRNA at the ribosome, and it leads to adding its specific amino acids to the growing polypeptide chain. And this process we refer to as the translation process. So protein synthesis, mainly we divide them into two: transcription, which is happening in the nucleus, and the translation, which is happening at the cytoplasm. And with the transcription process, DNA information coded into mRNA. The spelling, you have to have lowercase here, M, and then the uppercase letters over here. And the mRNA then decoded to assemble the polypeptides in the translation process. So let's look at them a little bit. The transcription, it's a process of transferring code held in the DNA gene base sequence to a complementary base sequence of the mRNA, and lots of transcription factors involved with, and they activate this transcription process. And it involves 3 steps: initiation process, elongation process, and the termination process. So what will happen with the initiation process is the enzyme, like the DNA polymerase, here it's RNA polymerase enzymes, it separates the DNA strand, because at that, even the protein synthesis, the DNA is twisted, so you have to open it up, the particular gene segment. And then The RNA polymerase, it adds the complementary nucleotides to the growing mRNA, which match the sequence based for the dRNA template. So if you have A in the DNA, you add the U for the mRNA. If you have G, then you have C adding. So that's what is called the making the complementary mRNA strand. And you have like a short 12 base pair segment where the DNA and mRNA are temporarily bonded at that time during the elongation process. So it's a temporary bonding between DNA and RNA. We call it as the DNA-RNA hybrid. But once it's finished, it won't last longer. And then the termination process, the transcription encounters a terminational signal in the DNA strand, and then you stop the transcription process, or we call them as stop codons. So here, what it's been shown here. So this is the coding strand, and what will happen is you have the promoter region, which has the start point, and you have the terminational signal. So when the RNA polymerase comes, it will open up the DNA strand. It will start to make the mRNA stand for the template strand, and it will make the growing mRNA strand as the RNA polymerase moves towards the terminational signal. And once it's encountered the terminational signal, you would stop the process of mRNA strand making. And this shows the bigger picture over here. With related to the processing of mRNA, mind you, if you remember, the DNA strand does have exons and introns. So during the transcription process, no matter whether it's an exon or an intron, it transcribed the whole DNA segment of that gene. And then what you need to do is you have to keep the extrons, but you have to remove the introns. So that is what is happening during the mRNA processing. So the mRNA strand is going to edit, and it removes the introns through a structure called spliceosomes, and the process we called as splicing. So at the end, the processed mRNA would have only the exon molecules, and that's the one which is going to go to the cytoplasm for further processing for the translation process. So during this translation process, when you think about translate, means you know one language, and if you want to translate into another language, then you have to convert it. So the same way, now it's in the nucleotide format. mRNA, even it's RNA, it's a nucleotide format. The protein is in the amino acid format. So now what we have to do is we have to translate the nucleotides into amino acids. So it's like converting another language. This process involves several structures: mRNA, genetic code, tRNA and ribosomes, translating events, and sometimes the rough ER. So let's look at them at one by one. So what does it mean by genetic code? So when you take the triplet code, remember the DNA has a triplet code, and it represented by complementary 3-base sequence on the mRNA called the codon. So you have 4 nucleotides. The codon involved only 3 nucleotides. So what would be kind of the possible combination, which is like you have 4 and you have 3 places. The possibilities would be, there could be 64 possibilities of these nucleotide combinations. And of these 64, there are 3 stop codons, that is only 61 gives rise to amino acid formation. And out of those 61, only 20 possible amino acids are being formed. So, which means like, okay, now you have 61 codons, because 6 out of 64, 3 codons will be stop codons. So they do not give rise to amino acids. But 61 codons have to code for only 20 amino acids. How could this happen? It could happen either some codons would not give rise to amino acid, but that's not possible. All codons 61 would give rise to amino acid. So then maybe 3 or 4 codons would give rise to one particular amino acid. So that process, we call it as the redundancy. And the redundancy is helpful to prevent the transcription errors happening during the protein synthesis. So this is what I meant. This is the genetic code. You see these codons, it's a triplet, UUU, UUC. So these two codons, these triplet codes, two triplet codes, code for the phenylalanine. These 4 triplet codes code for the proline. So even though there are 61 here, you only code for 20 amino acids. That's what it means. And these are the UUAA, UAG, UGA are the stop codons, and you would see there's no amino acids being attributed to those codons. And we mentioned that ribosomes are involved with the protein synthesis, the translation process. So ribosomes, they coordinate coupling of mRNA and tRNA. And ribosomes, they contain one binding site for the mRNA, and they have 3 binding sites for the tRNA. So the 3 binding sites would be the amino acid site for incoming amino acyl tRNA, which we call them as the A site, the peptidyl site for tRNA linked to the growing polypeptide chain, which we call as the P site, and the exit site for the outgoing tRNA, which we call as the E site. So like the transcription, the translation also have 3 processes: initiation, elongation, and termination. And during the initiation process, it happens when 4 components combine at the P site. So here you would see the P site of the ribosome, a small ribosomal unit, and the initiation tRNA carrying the amino acid methionine, and the mRNA and the large ribosomal subunit are the 4 components. And once these 4 components comes together, the next phase goes with the elongation process. So during the elongation process, the amino acids then added one at a time to the growing polypeptide chain, and this has 3 repeating steps, which are the codon recognition, peptide bond formation, and the translocation. So under elongation, these 3 processes would happen. During the codon recognition process, the anticodon of the incoming tRNA, it binds to the complementary codon of the mRNA in the A site of the ribosome. So the first one goes to the P site, the second one comes to the A site. And then in between these 2 amino acids, they form the peptide bond formation that is happening in the peptide bond formation process. So the amino acid, the polypeptide chain is transferred into the A site amino acid because it's formed the peptide bond. And the translocation process, the entire ribosome translocate, shifting its position 1 codon along the mRNA, that it moves in this direction. So that's the translocation process. And the termination process is where they encounter the stop codon, the entire process is going to stop. The rough ER involvement with the protein synthesis is like these amino acid segments, they have something called the ERN signal sequence. So they present on the growing polypeptide chain, and from the ribosome, these signals led this polypeptide chain to be docked into the rough ER surface. So we call this signal recognition particle on the ER, and that directs the mRNA ribosomal complex where to dock. So once it's docked, the forming polypeptide enters the ER, and then the sugar group is going to add it to the protein, and then the polypeptide chain's shape is going to altered, and this protein is now enclosed into the vesicle for to transport into the Golgi apparatus for further processing. So this is what it's been mentioned. So the signal recognition particle is existed, and then it's going to be docked into the ribosome. Ribosome is the rough ER, which has been showed in blue, and through this SRP segment, the polypeptide get into the ER, the sugar components is going to be added, and they're going to be packed into vesicles and then transported towards the Golgi. So with relate to overall this whole chapter, there are clinical relevance and importance things we need to discuss as well, and one of the things would be the apoptosis, autophagy, and proteasomes. So, so when the cell, they don't live forever and they do not live as smoothly, they do encounter for certain damages. And when they encounter any damage, they needs to be taken out from the system. So there are several ways of happening. One is the autophagy, that is like self-eating. In this process, there are non-functional organelles and cytoplasmic bits are being removed by the formation of autophagosomes, and they are going to be degraded by the lysosomes. And the unneeded proteins can be marked for destruction by proteins called ubiquitins. So what will happen is the proteasomes, they disassembles these ubiquitin tag proteins, and they do then disrupt these proteins, but then when you destruct, you break down them into their basic subunits, like which is the amino acids, but you don't destruct the amino acids, you can reuse these amino acids back in again. So that's why it's called like recycling the amino acids, and you can still use the ubiquitin as well. And with related to the apoptosis process, we call it as the programmed cell death. Unlike the autophagy, it's when the cell is dying that it's been going to be programmed, and certain cells do this, undergoes apoptosis, such as the cancer cells and the infected cells. Some old cells, they do the self-destructed. So how this happens is when the cell starts to die, the mitochondrial membrane starts to disperse, and there are chemicals is going to leak out, and these chemicals would activate some enzymes called caspases, and caspases cause degradation of the DNA, the cytoskeleton, and that leads to cell death. And the dead cells then being phagocytes by the macrophage cells in the body. And with related to the cell aging process, there are several theories have been put out: wear and tear theory, and the mitochondrial theory of aging, and the immune system disorders theory, and then the genetic theory. And genetic theory would be the which is much more accepted or kind of little proven one, because it says like cessation of the mitosis and cell aging are programmed into genes. So in every time when a cell divides, there at the end of the chromosomes, there are segments called telomeres. They get shortened up. So the more the cell undergoes division, it gets shortened out, and then the cell starts to age. The lifespan is going to shorten up, then we call it as aging. But there are enzymes, research have found, to lengthen these telomeres. So in that way, you would shorten or you delay the aging process. So this is the end of chapter 3, and thank you for listening.